people. Sorry about the accents. I'm actually from Liverpool in the UK, so it's not a very nice British accent. So yeah, I'm in my second year of PhD study, so I'm just going to talk to you a bit today about what I've done so far. So first, the aims of the study. So what I'm looking to do is develop a mathematical model for predicting novel regions in the genome that may potentially harbour genes that are associated with longevity that we're not yet aware of. And as I said, I'm going to be looking at using Drosophila to do this. The main thing I'm going to be using is network approaches. So what we're taking is the knowledge that we have of the 3D structure of the genome, as well as genes that we also know to be associated with longevity. And we're going to do this by integrating 3-dimensional chromosome interaction data with 2 GWAS studies. So we make 2 hypotheses in this study. The first is that this 3D structure of the genome governs the collocation of longevity-associated genes or genomic regions with novel regions that may have an effect on longevity. And the second is that SNPs residing in these regions might influence longevity either on their own or have a cumulative effect. So as I said, we're going to be using networks. So what we've done is created networks in which the nodes represent 80 kb regions of the genome and the edges represent connections between these regions. And once we've created these networks, we're going to be applying network measures to see which are the regions that we should be looking further into. So the first of the GWAS datasets that we've used is from a paper by Burke, and what this paper provides us with was SNP positions along with 8 young and 8 old haplotypes. And these, you can see here, these numbers here, these represent the proportion of how often these SNPs occur in these haplotypes. And what I did here was calculated what I've referred to as a d-statistic. And this was the Euclidean distance for these SNPs in 8-dimension space. So the second GWAS dataset was again a list of SNPs, but each of these SNPs was provided with a p-value which signified the significance of each of these SNPs in terms of longevity. So the lower the p-value, the more associated this SNP is with longevity. So I created these networks for each of the data sets. So first of all, we used nodes which had, so as I said, we calculated the d-statistic for the first data set. So we took those d-statistics greater than 7.9, which was a genome-wide significance level of 5%, as you can see in the first network. And then we used Hi-C data to add in connections between these nodes, which fell in the top 1% of the entire Hi-C dataset interactions. And we also then added additional nodes, which were regions which weren't found using this d-statistic value, but also had interactions with the original nodes in this highest 1% of interactions. And then we did the same for the second data set, except obviously these came with a p-value, so we used a p-value cutoff of 3.33 times 10 to the minus 5, which was an altered p-value that we calculated separately. And again, we added additional nodes. So what we then did was, as I said, we used network measures. So for example, here we've got the PageRank measure for the networks. And we then ranked these PageRank numbers from highest to lowest and looked at the top 10% of top 10% of regions using this PageRank measure. We then, in these regions, we then took all genes that resided in them and did go-term analysis on these genes. So what we were looking for was where these genes fell. So we've got obviously genes, we can see in the table, the genes under this go-term analysis that were found in novel regions of these genes here. These were the nodes that weren't original nodes. And we've also got genes that fell in the original nodes that were previously associated with longevity using a database in the GenAge database that you can see in the top right-hand corner there. So really, the original nodes aren't really of interest to us, as these have been indicated as significant by the GWAS datasets that I've used. But what we want to see is what is actually in these novel regions that are also interacting with these regions that we found using the datasets. So these were the results for the Burke dataset, the first dataset, and we tried to look at those that we might see to be significant in terms of longevity. And then we did the same for the second dataset, and as you can see here, we actually found Go terms very related to longevity. So we had aging, for example. Again, we found in the original regions, we found genes that were already associated with longevity, and then we found many more genes in the novel regions, and one of these was already associated with longevity. So what we then did was looking at both GWAS datasets. So obviously I talked before about using the page rank, but we also looked at the clustering coefficient. What we looked at was any regions that flagged up that were common between the both datasets. So we can see here, those in red are those that are common between both datasets and were also that were also new regions to both networks. So we've got 6 here, and then we also found there was more regions in common between both datasets using PageRank, but only one of those was actually novel to both of the datasets. So we then looked at all genes that resided in these regions. So for example, we found using clustering coefficient that there were already genes that were associated with longevity, so ESG and CMA5C, also ones with phenotypes that may be associated, so increased mortality. We did the same for the PageRank region, the one highlighted in red, and there was just one gene there that had some sort of association with longevity. So as I said, we're looking at SNPs. So what we then did was we looked at how many SNPs resided in these common regions between both bins that were also novel. And there were a fair number of SNPs residing in each of these regions. And then we also found that for this one region at the bottom, which was this PageRank region here, that had the most SNPs residing in it in the whole, like it had the largest amount of SNPs residing in it in the whole of the SNP data set. So we explored this region further, and we found that lying in this region of the genome, this secrepin family, so SEC-A1, SEC-A2, I'm not sure if you've heard of them before, but these were genes that are already known to play a role in immune response. So yeah, so this is what I've done so far. So it's just kind of indicating regions that can be explored further by network analysis, given what we already know. Thanks.
These are 5-minute talks, so we are not going to be able to take questions, but please catch up with the speakers afterwards. So our